at some, moving on from looking at how we get DNA making proteins, we're going to have a look at DNA itself. We're going to have a look at some genetics, and we're going to look at some of the things that basically go wrong. And it's only by looking at the things that go wrong do we actually learn how genetics actually work. So let's get down to it. What we've got is this idea of DNA replication, and it's very clever in the way that it works. We've got our two strands of DNA, and we've got our bases, A with T, C with G, T with A, and G with C. So we've got these bases, and each 3 forms a codon. It forms, when gone into mRNA and through the ribosomes, a amino acid, and this codes for a particular amino acid. And we have just ATCG going along here and so on, and that would have its corresponding base pairs, and when my DNA comes in and is copied, because we have semi-conservative replication, in other words, this strand is kept and this strand is kept, and a new strand is made with this particular sequence, then these will be exactly the same. So this is how DNA replication goes ahead. So when each cell divides for either reasons of growth or maybe repair, we need an exact copy of all the DNA. You started off here simply with one cell, a zygote, and that has managed to get to you as a complete being, let's say here an adult, with, I don't know, 20-30 trillion cells. I don't think anyone really knows exactly how many, but it's of that type of order. That number of trillion of cells, and all of these are identical. And this means that when the DNA copies, it's very precise in the way that it copies. Every time it copies, it doesn't make a mistake. Now, all this work is done with a particular enzyme, and this enzyme is called DNA polymerase. Right, now DNA polymerase is an interesting enzyme, and what that does, it's going to take the new base, and that is going to exist as a base, it's going to exist as a sugar and a phosphate. So we've got a different base. We've got 4 of these: A, adenine, thymine, cytosine, guanine, connected to a sugar, and for DNA this is deoxyribose, and a phosphate, and these are joined on one at a time. Now when they join one at a time, we can see that it's going to work in an odd way. So let's look at what I've got. I've got 2 strands of DNA I want to make, and I've got here my A, T, C, G, carry on the same sequence. Now as I want to make that, what I've got to do is put on first of all one base, and then I can link to this sugar phosphate. So I've got my sugar, if you like, with my phosphate, and I bring in a new one of these with its sugar and its phosphate, and we're going to join those together, and the enzyme does that. And as it joins all of these together, it's moving nicely in this direction. But how about if I've got a different chain, the other side? What's going to happen with that chain on the other side? Well, the enzyme's going to work, but it's got to work in the same direction. So if I split my piece of DNA, on this side it can go in this direction, but on the other side it's got to go in the other direction. And so it starts at different points and it moves along and joins up. So it's working one way in one direction and the other one in the other direction. Now let's explain this semi-conservative replication. How do we go about knowing that we've got semi-conservative replication? Well, we've got to do some experiments to try and prove this. Now there were a couple of guys, Messelson and Stahl, and they played around with some ideas, and I suppose the best way of trying to do this is to try and draw this out for you. And I've just decided I'm going to use some coloured pens. Right, let's get some coloured pens out so we can have a look. I've got a piece of DNA, and let's make it a nice pretty pink. So this is my piece of DNA, and for everyone who thought I was going to draw a nice DNA spiral, think again. I'm not very good at doing this. So we grow this in a particular medium, and we're going to try and grow it in, this was normal DNA, and they grew it with ordinary nitrogen. So this is N14 nitrogen, and there's a lot of nitrogen in here. Now this was grown, bacteria grown in here for a considerable amount of time. There we are, you can see it better. Now they took this medium and they grew these bacteria in a new medium which contained much heavier nitrogen. So this is going to be nitrogen 15. It's got an extra neutron. And they were looking for 2 ideas: whether they were going to get the new strand made of 15 to go with the old strand, or, and that is this semi-conservative, oh, can't spell, replication. Or could we have a different idea where this formed a template and we'd end up with a new double strand plus the old strand. So what we'd have assumed we got was either this system or that system. And in fact, we found this one. So what did they do? Well, Messels and Stahl basically took their bacteria that was growing in normal nitrogen, and this meant that basically every time it replicated, we've got ordinary nitrogen. When they put it into a medium containing the heavier nitrogen, then it grew for a little while and they basically stopped the reaction, they killed the bacteria, and then they put it into a density gradient solution. Now let's see what they would have expected. And I've got all my pens all caught up. Right. Now, if I just take my test tube and I centrifuge it, then we get our light DNA forming in one place. And because it's light, it's going to be higher up. So there's my light DNA. When we looked at the stuff grown in the heavier nitrogen, what they expected to find was the light strand, which would come from this one that was still available, plus these two, which would be a medium strand, a medium weight, because these are made up from one light plus one heavy. If the other system had worked, then they would have expected for this to get from the beginning one a light and a light and a heavy. What they also expected to do was, if they left this to carry on, then what they would expect to find is the light continuing, get a medium, and then getting a heavy. Huh? What's going on? Well, let's look at what's happening. I've got this system. I've got these two genes, these two chromosomes, if you like. When these replicate, and they're now only replicating with the dark color, then what we're going to get is this set left, and what we're going to get is this one, these strands splitting, so that will go with heavy, and this one will get a light with a heavy, and on this one we'd get the light with a heavy and the heavy with a heavy, and the light will actually disappear. They actually found that the light carried on because some of the genes didn't actually copy. But after a while, what they did find was they only had medium and heavy, and after a while even that light gradually disappeared, leaving them only with heavy. So this little experiment did prove that we've got semi-conservative replication, and semi-conservative replication is not a very unobvious way of trying to do it. It's the way that if we talk to most students, they'd expect it to actually work. Now let's go on and have a look and see what's going to happen about mutations. So let's look at mutations, because it's only with mutations we can actually see how things work. So I suppose the first question is, what causes mutation? Do they happen normally all the time? There we are. Now, various things can cause mutations. I pop outside and we meet ionizing radiation. This is from the sun, this is from cosmic rays, this is from the ground, this is from some of the air, and basically what this is going to do, it's going to break either one or both DNA strands. And generally there is repair for this available. Right, the next thing that we can have is something called a deaminating agent. This deaminating agent is sort of a chemical change, and this chemically alters the bases in DNA, converting various things. And one thing it does do, it can change cytosine to uracil, and that can change the base sequence. We'll look at that in a moment. We can have alkylating agents. Now these alkylating agents are going to add methyl or ethyl groups that attach to the bases, and this gives us incorrect pairing. Another one we can do is we can meet base analogues. These get incorporated into the DNA in place of the usual base. So for some reason an A gets changed to a C, or a T gets changed to a G, or any combination of these things. And our favourite one, viruses. And a virus may insert itself somewhere into the genome to try and hide, and that may just add a few extra base sequences in somewhere. So this is just going to change the sequence. And all of these things are going to cause a change in the DNA. Let's look at each of these in turn and we'll see what's actually going on to see what happens. So let's have a look at ionizing radiation. So I've got my groups here, A, T, C and G. Radiation comes along, zaps that one out. If we've got the bases as they were in its sort of double form, then we can repair this base by looking at what we've got here and saying, well, we've got a G here, so that must have been a C. If, however, this is taken out and the opposite one is also taken out, then we may know we've got to replace this with a base, but it can be replaced with one of 4. And it might replace it with a C and its complementary base, the G, but it may not, and it may produce something else. And what I can do is show you a little table which I alluded to last time, which might help. I'm just looking at a book with one of these genetic tables in, and this is looking at the mRNA codons. So if I've got a U followed by a U followed by a U, then this is going to give us phenylalanine. If I change the last base to a C, it makes no difference. I still get the same amino acid put in place. But if I were to change this to an A, just this last one, we change it to leucine. If we change it to a G, we change it to leucine. Some of them aren't going to make any difference. Changing the last base here, UC to a U, C, A or G, is always serine. These are always proline. These are always threonine. But other ones can cause bigger problems. So changing in here, changing just the one base can change this. If we change the middle base, then much more likely to have a change. And if we change the first base in the sequence, again it's much more likely to cause a change. So we've got quite a few of these different ones. If I push this up, you can see there is a list of what all the different bases are and what they stand for. Sometimes in an exam you might be given a table like this and have to process the information from it, but that isn't today. So ionizing radiation is going to change one or both strands, and when we've got this, we can either end up with a replacement, which is just basically a substitution, or we could lose the base altogether, and that causes interesting problems. If I have a replacement, for instance, and I'm just looking at the RNA here sequence. If we go from UUU and we go to UUC, we're not seeing any change, because that is phenylalanine going to phenylalanine. If, however, UUU was substituted with something like UUA, then we are seeing some changes, phenylalanine to leucine, and the properties of these are very, very different, and because they're so different, they cause big problems when we translate this into the protein. But what happens if we have a complete loss of a base? So if I've got here AT, GC, ATGC and ATGC, if I lose this base and it's lost completely, then I've got ATC for my first one, and then ATG for my next one, and CAT for my next one, instead of having ATG and CAT and GCA. And we've now got an amino acid completely changed here, because the sequence, although we've just moved out one base, the whole sequence has changed dramatically, and this does cause significant problems. Let's look at an example case of this. So let's just change one of the bases, and changing one of the bases is a disease that we meet called sickle cell anemia. Yeah, not very good spelling at the moment. Right, sickle cell anemia. Now, what is sickle cell anemia? Well, we're looking at one base change, and it depends where this one base change occurs. So what we get is a GUA rather than a GAA. Looking at my little table, what does that do? So let's look at my little table, GUA, GAA. So GUA, valine, to GAA, which is glutamic acid. So valine changed to glutamic acid. These amino acids look remarkably different. They are charged differently, and so instead of having the non-polar valine, you get polar glutamic acid at this point. This small change has got an absolutely devastating effect on the functioning of the molecule. It makes the hemoglobin less soluble, and when oxygen levels are low, these molecules form long fibres that stick together inside of the red blood cell, and this distorts its shape, and the resulting red blood cells, instead of being round, are sickle shaped, and they carry less oxygen, and they're well known for blocking capillaries, causing all types of problems. But there's one good thing. So these people can't run very well, they can't survive terribly well, and I'm saying there's a good thing, yeah. And the good thing is that they don't get bitten by mosquitoes, so they don't get malaria. So everyone else dies of malaria, and these people, although poorly, survive. And although they survive, not terribly good, but they're better than the ones that are dead. And so sickle cell anemia exists in about 11% of the population. Do we see these things occur in lots of other diseases? We certainly do, and one of the most popular ones that's studied is cystic fibrosis. And that is also due to a change in normally one particular base pair. There are in fact lots of different types of cystic fibrosis, and they do have different changes, but the basic idea behind this mutation is if I've got a protein that does that sort of shape and I affect one of the bases here, then I can radically alter the shape of the molecule. This means that this protein basically won't work. So we've got these different agents causing different problems. We've got the problem of a deletion of a base, and we've got the problem of a translocation. Of the bases. And generally these things aren't terribly good. We can get duplication of some of these sequences, and if the wrong strand gets broken off and put in, then we can get inversion of the base sequence. So we've got the bases being changed, and then we can have chunks of bases being changed. Generally, these are going to cause problems. But very occasionally, we get some beneficial mutations. I'm not really talking about here our Incredible Hulk, someone getting exposed to radiation and becoming sort of very different, but there are some cases where we get this beneficial mutation. Now, the ability to digest lactose, which is the sugar in milk, is basically thought to be the result of a relatively recent mutation. The majority of mammals in the world become lactose intolerant after they cease to suckle. And basically the ability to digest lactose is found primarily in the European populations, and they're the ones that are more likely to farm cattle. So the ability to drink milk and process the lactose as an adult can help prevent diseases such as osteoporosis. This mutation seems to have arisen sort of spontaneously more than once, and it's also found in various other people who live in East Africa. So there are occasionally beneficial mutations, so something that would disable an enzyme this sort of to play around with, digest lactose, probably lactase, is a useful thing. It can happen, but generally beneficial mutations don't occur very often. Sometimes we get duplication of genes, and one of those cases is looking at someone who's got colour vision. Now, if you've got colour vision, you've got 3 genes. You've got 3 very similar genes that do very slightly different things. So I've got one gene that, say, can read red, one that can do green, and one that can do blue. Originally we started off with just one of these, and for some reason this got duplicated, so I've now got 2 red genes, probably somewhere different in the genome. Well, that's no problem, because I can either use this one or this one. But this one now suffered a mutation, and what's happened to this one is that the frequency of light that it can receive is now gone from the red to the green. And now, if I've got both genes, I can see more colour. And maybe I've got that green gene, and it undergoes later on a duplication, and we get a mutation, and then it can see blue, and we've got some beneficial mutations going ahead. I'm told that some snails have 5 versions of these genes, and they can see even more colours than we can. As to what those colours are, I'm afraid you're going to have to ask a snail, because I don't know. I'm one of these poor souls that's got one of these very slightly mutated, which makes me colourblind. Which always gives rise to the interesting question which students ask me is, well, what colour do you see? And the answer is, well, as far as I know, I see things quite normally, because I can't see what they see. So we've got DNA changes, we've also got chromosome changes, we've got these deletions. I can delete a whole gene. I can move a gene from one chromosome to another one. That's not a problem, but it may be that only part of a gene gets changed, and this can give rise to maybe beneficial mutations, but usually not. We've got duplication. Now this means you're carrying around a lot more DNA than you need. And if we look at the human genome has quite a lot of duplication, and you're carrying around lots of spare bits of DNA, which maybe one day might become useful to you, or maybe you're just carrying around a lot of wasted DNA. I can't see that really ever that inversion is going to be useful, but it might just have a little bit of serendipity and produce some sort of beneficial mutation, but probably not. When we have meiosis occurring, then this is where these happen, and this gives rise to new sorts of offspring, and with these new mutations we are looking at probably trying to sort of produce a new advantage to a creature. They may be useful, they may not be. So that gives us some idea about how these genetic things work. Now, how does it really work? Well, let's finish off by having a look at something called the Lac Operon. Now, you'll find the Lac Operon if you want to watch a simulation if you go to fet.colorado.edu, and you'll see there a Lac Operon simulation, and very good it is too to look at. Basically, the idea is very simple. So what we've got is we've got a gene. So let's just draw out my gene. So here we've got my gene, and what I've got to have here is something called basically a promoter. Now, this promoter is where we're going to actually fit something like our RNA polymerase on. And it goes on, and it then moves along, and it produces a copy of this gene, and we turn it into mRNA. But I do have an extra little piece of DNA here, and somewhere else in the gene, in fact just a little bit further along, is another one of these little promoter sites, and it produces a different gene. We'll do it in a different colour. So my RNA polymerase goes on here, it binds to this promoter, and having bound to this promoter, it goes along and it produces a little protein, and this little protein, what it does is it can stick to that piece of DNA. If it sticks to this piece of DNA, this RNA polymerase can't move along and can't produce this gene. So this gene is effectively blocked and it's not made. If we've got, however, any lactose present, then this little gene here has a lactose molecule bound to it. And when it does that, this changes shape. As it's changed shape, it can't now bind to this gene. And if it can't bind to this gene, then the RNA polymerase can go along and it can make here the lactase, which can break down the lactose. And it does so, and it keeps breaking down the lactose until there's no more. And then this now, no lactose, the lactose can't bind to this inhibitor gene, so it doesn't bind, so this is now in existence, so this can now bind to this site on the lactose gene, and when it does so, the RNA is blocked, RNA polymerase is blocked, so it's not made anymore. So this is quite an efficient way of having feedback, so that a small gene is being made and produced all the time, but it's stopping a big one being made. And this is quite efficient in the way that the body works. So this is the Lac Operon. We've got this little repressor protein that's made, but this repressor protein can itself be repressed by the binding of the lactose. So this is the way that we get some sort of transcriptional control over the way that genes are used, and this is just the way that the Lac Operon works, but we get some other equally exciting ones, but generally for A-level this is the only one that you need to know about. So it's this idea of controlling everything. Now, can we control things any other way? Yeah, we can open up genes by binding to them, and this is the topic of epigenetics, and this is what we're going to look at next time. So until then, I hope you've learnt something. I hope that's been useful, and if you have found it useful, then by all means subscribe, and I'll see you next time. We got any questions on any of this? No? Okay, so I'll see you then. Take care. Bye bye.